Hi everybody, welcome to lecture number 3 in our evolutions unit. So we're going to talk today about the evidence of evolution. And there are 6 scientific disciplines that you need to be familiar with. The first is molecular biology. So this shows us that there are molecular homologies, which are similarities in molecules between species. The second one that we look at is anatomy, and that shows structures that are due to relatedness, so they're similar. The third is embryology, and this is where the study of embryos shows similarities in development. The fourth is paleontology, and this provides fossils, and this is going to help us determine the history of evolution. The fifth is biogeography, and this is the study of distribution of species in different regions, and this supports natural selection. And the sixth is mathematics, and this helps us by providing a way to estimate ages of fossils to determine the history. So what evidence do we have to support the idea that species have evolved? So the first one that we're going to look at is going to be molecular homologies. And this basically is like saying the similarities. So the molecular homologies are similarities in the molecular or cellular processes of different species. So here are some examples. All organisms use the same 4 bases in their genetic code. They all utilize DNA, RNA, and ATP for energy. They also all use the same 20 amino acids to make proteins, as well as the same codons that code for the same amino acids. And they have the same wobble effect in the genetic coding of the amino acids. So for example, CUU and CUG both code for leucine, so that's the wobble effect. We also have similar proteins found in all living organisms. So for instance, cytochrome C. It is found in the electron transport chain of all organisms. We also have ATP synthase, which is used to make ATP in all organisms. And then we also have hemoglobin. And this is a protein that is found in many species, but not all of them. So if you look at these diagrams here, this is what the amino acids reveal. There's not a lot of differences from humans. For instance, we have no differences from chimpanzees, versus the less evolved you get, the more differences there are. So for instance, yeast, there's 42 differences. So all organisms are going to contain ribosomes that produce our proteins. And here are some examples that occur in different organisms. So all organisms utilize the same 2-step process of transcription and translation, by which information in the genes is going to flow into proteins. So again, going from DNA to RNA to proteins. And again, remember, this is called the central dogma. Also, all organisms perform glycolysis in order to generate ATP for energy. So this here is important for the AP exam. You must be able to list some molecular homologies found between species to help determine relatedness, and you must be able to use the data related to those homologies to draw conclusions. So for instance, looking at this graph, determine which species shares a most recent common ancestors to humans and justify your answer. So the first thing that you need to look at is most recent common ancestor. So basically that means which one is more related to humans. And in this case, it would be the gorilla. And we are looking at amino acid differences in the hemoglobin protein. So basically you need to be able to justify your answers that we share all of the same amino acids except for one. The second thing that you could be asked to do is to draw a phylogenetic tree to indicate the most evolutionary relationship between humans, gorillas, and the rhesus monkey. So in this case, if I can get my computer to participate, what you're going to do is you're going to draw a line, and right here at the beginning, this is humans, and then the more closely related to us would be a little tree off here for gorillas, and then we have 8 with the rhesus monkey, so we would have another branch off there for the rhesus monkey. Okay, so let's look at an example AP problem. So iron is an essential nutrient that's acquired by organisms from the environment. When the cellular levels are relatively high, the living cells synthesize an iron storage protein called ferritin. The induction of ferritin synthesis by iron was investigated in rats, and this is the results of that. And the proteins were measured in the presence or absence of iron and actinomycin D, a drug that inhibits transcription. And figure 2 is showing the results of that. So the gene sequences responsible for the iron-mediated changes in ferritin protein levels are highly conserved and are called iron response elements, or IREs. IREs have been observed in a number of genes involved in the iron transport and metabolism. The IRE sequence found in the ferritin gene are found in all multicellular organisms, whereas other non-ferritin IRE sequences are found only in certain vertebrate organisms. Which of the following is the best explanation for the observations? And then normally it would have, multiple choice and everything. But the point here is that it tells you right here they are found in all multicellular organisms. So what that tells you is that it's going to indicate a common ancestor. Okay, so evidence number 2. This is the homologous structures, and I cannot spell, I'm sorry. So homologous structures are anatomical body structures that are due to a common ancestors, and sometimes these are referred to as anatomical homologies. I don't know why I can't talk today. All I can do is apologize. So the following are examples of these homologous structures or anatomical homologies. So for instance, the flipper of a whale, the leg of a cat, the arm of a human are all homologous structures. So the conclusion is that the similar limb structure in different species is going to indicate a common ancestor. So you can look here and you can see this is a human. We've got these 2 bones and we've got those 5 fingers or phalanges there, right? Same thing in the lizard, same thing in the cat, same thing in the whale, same thing in the bat. It just, goes on and on. So Again, you must be able to list examples of homologous structures in different species and be able to use the information to draw a conclusion about relatedness. So, for example, use the information above to determine and justify the species that is more closely related to dolphins. So here, if we're looking, so we've got a human, a shark, a penguin, and a dolphin. So automatically, I think that we tend to lump things together based on where they're found. So for instance, the ocean, your first instinct might be the shark. But if you look, it's completely different than the dolphin's flipper. And the penguin is kind of similar, but the most similar would be the human. So your answer would be humans due to the homologous structure of the fin compared to the forearm. And you could even go so far as to list the 2 bones and then the 5 fingers type structures. Another example, which of the following pieces of evidence best supports the hypothesis that birds and crocodiles and alligators are more closely related to each other than they are to other organisms? So in this case, you have to be able to determine what type of evidence is going to be most useful. So let's look at these. So modern birds and crocodiles utilize different niches within the same geographical region. So that is no. Like turtles and snakes, modern birds and crocodiles build nests in which they lay shelled eggs. That's not really going to help us determine relatedness because a lot of things lay eggs. The skulls of birds and crocodiles have an opening that is absent in the skulls of other reptiles. That's a contender. Fossil evidence indicates that both modern birds and crocodiles originated during the Jurassic period. That doesn't help us determine relatedness, so I'm going to go with C. Because that hole in the skull, just like the bones in the dolphin and the human, is going to show a level of relatedness that all the other options do not. Okay, here's another example. Lobed fin fish were present in the oceans of the world approximately 400 million years ago. The first tetrapods, which are vertebrates that had limbs and could move on land, date to about 365 million years ago. One hypothesis states that early tetrapods evolved from lobed fin fish. Which of the following is the best plan for testing the hypothesis? You must be able to determine what type of evidence would be most useful to determine the relatedness. So, determining the number of fins found on the lobe fin fish from different geographical locations. That doesn't really help us. Measuring the proportions of radioisotopes found in fossils. That requires a date, so that's not really going to help us in this instance. Comparing the arrangements of bones in the fins of the lobe fin fish and the limbs of the earliest tetrapods. That could be possible. Sequencing DNA isolated from fossils and the size, help to determine the size of the genome. Again, we're not really looking for fossils. Looking for DNA in ancient fossils to determine, again, ancient fossils. So we are going to go with C, that arrangement of bones. So sometimes your best option is to determine the ancestry of the relatedness by looking at the structures in fossils because they will be preserved. However, DNA gets destroyed over time. So DNA is usually never a good answer. The oldest DNA recovered was from plants, and it's about 500,000 years old. And in the most ideal preservation conditions, scientists estimate that every bond in a DNA molecule would be broken down at 6.8 million years. So that sounds like a long time, but you need to keep in mind that dinosaurs went extinct approximately 65 million years ago. So you can see why DNA is not ideal. Evidence number 3 is the vestigial structures. So these are remnants of functional structures that form in modern species, but they have no discernible function. So these structures suggest that the species evolved from an ancestor that did utilize those structures, but that modern species still have the genes that cause the development of the structure. So, for example, the wings on ostriches and penguins, they don't fly. So they don't really need them, but they're still there. The pelvic and leg bones in snakes and whales, so you can see here. But again, they don't walk using legs, so they really don't need the pelvis. And then the tailbone and the appendix in humans. We do not utilize a tail, and the appendix has no validated purpose at this point in time. Evidence number 4. We see similarities in embryonic developments between species. So we can conclude that those similarities in early development indicate relatedness and a common ancestors. So an example is, for instance, the pharyngeal pouches or gill slits. And you can see here in the fish, the reptile, the bird, and the human, they are present in every single one of them during embryonic developments. So in fish, they obviously develop into gills, in amphibians, and in humans, they actually develop into ear cavities. The 5th type of evidence that we have is transitional species. So this is going to offer physical evidence of succession by showing the changes over time. Usually simpler creatures are shown, and then it shows more advanced species later in time. And you can see how those transitional species, how they change, they show the change that took place from ancestors to modern species. So for instance, the evolution of horses, they evolved from ancestors that were much smaller and had 3 toes. And the transition species can help you see the changes that actually occurred over time, where they've gone from those 3 toes, those small creatures, to a single toe. We can also see in the evolution of whales, they evolved from ancestors that were mammals and actually walked on land. And you can see the transition and how it has occurred over time. Really quite interesting. So the evolution of birds also. Birds actually evolved from reptiles. And a transition species was discovered called the Archaeopteryx. And it shows the link between the dinosaurs and the birds. So it actually has reptilian features such as scales, but it also has wings and feathers. So dating of fossils is important because that allows us to draw accurate conclusions about the evolution. And they are dated 2 ways. There is absolute, and then there is relative dating. So absolute dating is used to determine a specific age of fossils or rocks around fossils. So for example, through absolute dating, we can determine that this fossil over here on the right is approximately 3 million years old. Versus absolute dating, it's going to use the decay of radioactive isotopes, and we can determine the age by determining the ratio of atoms like carbon-14 and carbon-12, because carbon-14 actually loses neutrons to decay, and for C12, it takes 5,730 years for half the C14 to decay to C12. And this is called the half-life of C14. So if we are asked how old is the fossil that contains 50% of carbon as C12 and 50% of carbon as C14, then it would be the 5,730 years old. How old is a fossil that contains 88% of C12 and 12% of C14? So it's about three times more. So as simple as it sounds, you would basically multiply that 5,730 years by 3. So it would actually be 17,190 years old. Okay, relative dating can only give us an idea of a sequential order of events. So for example, these fossils are older than these fossils. So that's basically all it's saying. It's going to say these were here before those. And they're going to do that in a chart such as this. You must be able to use relative and absolute dating methods to draw conclusions about the evolution of a species. So let's look at an example. The fossils of the lobe-finned fish, which are ancestors of amphibians, are found in rocks that are at least 380 million years old. Fossils of the oldest amphibian-like vertebrate animals with true legs and lungs are found in rocks that are approximately 363 million years old. 3 samples of rocks are available that might contain fossils of a transitional species between the lobe-finned fish and amphibians. 1 rock sample that is 350 million years old, 1 that is 370, and 1 that is 390 million years old. Select the most appropriate sample of rock in which to search for a transitional species between the lobe-finned fish and the amphibians and justify your selection. Well, they told us that the fossil of the lobe-finned fish are found in rocks that are 380 million years old, right? They told us that the amphibian-like are in the 363. And then the samples that they gave us are 350, 370, and 390. So obviously, it would be the 370. And you would justify that by saying 350 million years is after amphibians were discovered, and 390 is before the lobe-finned fish were discovered. So that transitional could not occur before or after those were discovered. Okay, and finally, evidence number 6, we are going to look at the biogeographical evidence. And this supports the idea of evolution by natural selection. So this one's important. Observations that support the idea of natural selection. So we've got studies that show organisms seem to originate in one location, and then they spread to accessible regions, and then they begin to vary slightly over time due to natural selection. And this here shows you with butterflies, actually from South America. Also, closely related species are usually found in areas that are geographically close to one another. So members have migrated and colonized new locations, and then they develop those different traits due to differences in the environments, as well as natural selection. And evidence number 7, we can observe the evolution of populations, so we know that the traits of populations change over time. For example, we have witnessed the development of strains of bacteria that are now antibiotic resistant. We have also witnessed in our time the rise of plant varieties that are now resistant to herbicides. And we have witnessed the rise of insect populations with individuals that are insecticide resistant. And also the Galapagos finches on the Galapagos Island, they were studied recently, and there is an emergence of a new species, and that has been recorded by researchers. So that brings us to the end of lecture number 3. I will see you guys next time when we start talking about our phylogenetic trees and cladograms.